Good, whatever time it is you're in. Thank you very much for joining us today or tonight. And hopefully this is our fifth and webinar series, and this one is about integration of EEG and fNIR. We have had a number of other webinars. Some of them have some overlapping content, so this one I will try to skip through some of the parts that may have been covered in previous ones. Or maybe covered in future ones as well. Thanks again for joining us. So today's, the outline for the talk is pretty straightforward. I'm going to talk about, introduce wearable dry EEG. I'll then introduce fNIR, functional near infrared, and I'll talk about integrating both of these together. So if you don't know anything about EEG, we're going to have a quick introduction for you. If you don't know anything about fNIR, there'll be a quick introduction for you. If you know a lot about both of them and are curious about how to put the two together, we'll have something for you. If you are here for the COVID webinar, that's the webinar next door. So with that, let's get started. For those of you who don't know us, we are Wearable Sensing. We have been revolutionizing brain monitoring over the past decade with our breakthrough dry electrode EEG technology that was developed by Quasar. And we brought, this has allowed us to bring brain monitoring into the real world for new innovative applications. And today we're going to be talking about adding fNIR onto that. So to start off the story, we will go really quickly into the history and talk about, introduce you to the early human circulation balance developed by Angelo Mosso. And the idea there was you put a person on a sort of a tipping table and you ask them to think deep thoughts or give them some challenging problems or emotional thoughts, and you watched as the blood flows into the brain. So sort of a very early functional near infrared. On the other side is, of course, the first human EEG recordings by Dr. Hans Berger in the early 20th century. So that's just a little bit of anecdotal history. There are a number of different brain monitoring technologies today. Typically, when we scientists look at them, we talk about spatial resolution and temporal resolution of these modalities. And we look at how well we can see inside structures, how deep, what's the time scale. But if we wanted to go into the real world, we have to think about other factors as well in terms of how easy is it to use, how available is this technology, how robust to artifacts, motion or electrical, how portable is it, how cost effective. When we look at these parameters, we can see that EEG and near infrared have a nice set of pluses going for them there. Of course, when we pay closer attention to the spatial and temporal resolution factors, we see that they're also complementary in that NIRS has good spatial resolution, whereas EEG has a really good temporal resolution. So they really complement each other nicely. And they measure different signals, and we'll talk about those. So first, let's introduce EEG. So EEG is the measurement of the electrical activity of the brain from the scalp. When enough neurons fire together in the same area, we can see their local field potential sum, and that summation can reach all the way to the scalp. We can measure that with electrodes. You rub the skin first to get rid of the dead layer of cells and some oils. You put the electrodes, you apply some gel through those the holes in the electrodes, and we can get measurements that look like this. So these are illustrations of the different types of morphologies that you can get in an EEG, depending on the state of arousal the person is in. So you can see an aroused state versus a relaxed state. You see those spindles here are called alpha waves. This is as the person goes to sleep, you can see different frequency of spindles. And when they go to deep sleep, you see these large oscillations that are called delta waves. Typically, so that you can see morphologically differences in the EEG that relate to the state, but typically when we analyze it, we look at the Fourier domain, we look at power and bins, and we give those names, delta, theta, alpha, beta, depending on the frequency range of each of those bins. So if EEG is so great and able to measure EEG signal, brain signals, what are its limitations? Why isn't it everywhere? Well, it requires some careful application of the electrodes. You have to rub the skin to bring the contact impedances down. You have to apply these gels. That's okay for a one-time session, but it can get painful, irritating, risk of infection, and really not enjoyable for people. You also need to have some good time synchronization for your EEG devices, something to give you context of what is happening when you're recording the data. Typically, EEGs have not been considered as really portable or ambulatory except for specialized devices, which were usually suitcase-sized devices. There were still lots of cables and motion artifacts until the advent of active electrodes. Still, they weren't really seen as a practical for real-world or virtual world applications. So this is where we came in. So I'm going to be describing to you the technology developed by Quasar, and as I'm describing that, on the bottom right, you'll be seeing a dawning on a long-haired subject of a DSI-24. So this has a headset that has 19 electrodes, and you'll be watching this recording, but it will be playing back in real time. So Quasar had a mission initially by DARPA, later by NASA, NIH, NSF, and various other government funding organizations funded different aspects of this technology development. The first mission was to take our high sensitivity, low noise capacitive electrodes that we had developed for cardiac sensing and other applications and deliver a dry electrode EEG. So we took those sensors, we added the resistive element to them, we added gave them some ultra-high impedance amplifiers, some custom circuitry, put some pins made of silver chloride, and we developed dry electrode EEG. The sponsors were, of course, very cautious and asked for us to validate that we were getting the same signal quality as we would get from a conventional wet electrode. And here you're seeing a recording of alpha activity simultaneously from our hybrid resistive capacitive dry electrode and the wet electrode. So you can see these traces here. We're getting better than 90% correlations, really good matching of the morphology. So we're pretty happy. Of course, the sponsor came back and said, yes, but we worry about artifacts. We hear that dry electrodes have huge contact impedances, and therefore they're going to be susceptible to electrical artifacts. People in the past had indeed tried dry electrodes, and when they put them on the head, because there's no gel to hold them in place, they can slide and create triboelectric artifacts, which produce large motion artifacts. So we did quite a bit of innovation on the electrode side. First, of course, the amplifier is immediately behind the electrode, rendering it an active electrode. There's some custom circuitry I won't talk about today. But we also put a Faraday cage. So this outer ring and this back metal plate create a Faraday cage, so a shield to protect the electrode and the amplifiers from all sides. The only remaining opening is the bottom side, which is where the head is and where, therefore, the EEG signals are going to come in. So this electrode and the amplifier are going to pick up only EEG signals, amplify them, and then send them out to the digitizer downstream. So that helps us deal with electromagnetic interference coming from the outside being blocked by the Faraday cage. To deal with the motion artifacts due to triboelectric movement of the electrodes in the hair and on the scalp, we patented this spring-loaded mechanism with a gimbal that allows us to have friction between the skin and the inner electrode, while if the headset moves, the inner electrode stays in place. So that provides some motion stability. At this point, so it's been 3 minutes, the dawning is done. The last thing you saw me do here, I was manipulating the electrodes through the hair of the subject. So we go through these rotation movements to get it through the hair. Okay, so this, we talked about how we got the sensors to get good signals with no artifacts, and then we spend a lot of time designing headsets that were easy to put on, comfortable to wear over extended periods. And then, of course, we added wireless data acquisition, both Bluetooth and onboard storage, so that you could go about and walk around freely in the world. Okay, so now that we've seen the dawning, let's take a look at this person's data. So first, we check the contact impedances, and we see everything is in green, so that gives, and a few other signal metrics, so it gives us a sense that we are good to go. Here, we are now seeing EEG traces. For those of you who are not familiar with EEG, each line is an electrode with relation to reference. Here, the references are linked ears. The top channels are the frontal electrodes, and those were some blinks. So those were 3 blinks that she did. And this is what her resting state EEG looks like. These are jaw cleanses, so she just bit really hard, and this is the EMG artifact spread through the skin into the EEG electrodes. That's a normal artifact we're showing here to show you the high frequency elements. Here she closes her eyes, so her eyes are closed, This is the alpha activity that gets generated when one's eyes are closed, mostly in the parietal cortex. So this is the default mode network, and being synchronized and activated at about 10 Hertz. Now we'll introduce this red line. So I'm tapping my foot near the subject, generating a lot of triboelectric artifacts, which you can see here in red line, but you don't see them in the EEG channels. Now the subject is tapping herself, her foot, and again, you see the EEG channels are quite clean, and there's no artifact removal here, no regressions of any sort. This is just a 1 to 40 Hertz filter. We just removed the 40 Hertz low-pass filter, and now she put her foot on the electrical cable, and you can see a lot of pickup of 60 Hertz frequency on this red channel, but nothing in the EEG. It's nice and clean. Again, we're not affected by these electromagnetic interferences. Lastly, we're going to ask her to get up and walk, and you'll be able to see her footsteps, the artifact of her footsteps on the red channel as she strikes the floor. Big, large triboelectric discharges, but nothing in the EEG. Very nice and clean signal. Again, the only filters that we have here are 1 to 40 Hertz filters. No other artifact removal algorithms. All right. So now that we've shown you the signal, so this is a quick iteration. We've shown this in the past. I'll go really fast here. You can see that in other webinars. This is an evoked potential, showing that the blue and the red, wet and dry, give you the same morphology and amplitude of the signals. And this is an SSVEP signal, showing again that we can get the SSVP signals in both the wet and dry. We're also able to record a seizure at one point on an infant, and we have this red line here shows that we have 6 times less 60 Hertz artifacts on our system. So that was a quick introduction to our technology. We currently have several headsets out on the market that change vary in different number of sensors, so it's full head coverage, smaller number of electrodes, the sleep wearable version, and the version that was designed for BCI, either P300 or VEP. And some of these have child-sized versions, and most of these can be used in virtual reality with different adapters we've developed for them. All of those produce raw data, either through CSV or EDF files, or in real time by streaming those to third-party applications. We also have a number of sensors that are complementary that plug in, such as ECG, MG, OG, GSR, respiration, temperature, and a built-in 3D accelerometer. And today we're going to be talking about fNIR, but we also will have another session about PECS. We've already had one about motion tracking and eye tracking. So what have I told you about electrodes? We have a high fidelity EEG sensor that's dry, artifact resistant, very fast to set up, comfortable to wear. You'll have to trust me on that today, and please contact Cameron afterwards if you'd like a demonstration a trial at your site, so you can assess the comfort and ease of use. So most of these are quite wireless and portable, and open up a wide range of applications in the EEG space. So that was a very fast introduction to wearable EEG. Now I'll move into fNIR. So thanks to the great vasculature in the brain and the fact that light will go through the head, we are able to measure functional near infrared. So as your neuronal areas get activated, they will use up some oxygen in the blood. So this will decrease the available concentration of oxygen in hemoglobin. The blood will quickly reactivate and re-oxygenate this area, send the new freshly oxygenated blood to the area, and so you'll see changes in the oxygenated and deoxygenated hemoglobin. So this is an illustration of that. So this is the basal state. As you start using as the brain area starts getting activated, you get less oxygen in this area, and that quickly gets repopulated with freshly oxygenated hemoglobin. So this is the time course of these deoxygenated hemoglobin concentration and versus the oxygenated hemoglobin concentration, where time 0 is the start. This is the start of the activated brain activation. All right, clearly the response curve shows you in time that the responses are a few seconds delayed compared to a neural response. So the EEG responses are going to be much faster than the buildup of the blood oxygen level density measurements. And there's also some evidence that shows that very, very small neural responses that may show some EEG may not evoke a measurable fMRI or fNIR signal. Also, the neural activity can saturate, so we'll max out, but the CBF can continue to grow. So there's some differences between these signals just by looking at the physiology behind them. I'm going to now illustrate how fNIR is measured. So this is an illustration of the scalp and the skull and the brain and the cortical areas. And this is a simplified rendering of that. But what you do is you put a light source and a detector. And as you shine light through, the light is going to scatter all around inside the tissues. It will go through the skull at near infrared. It will go through the skull, it will go through our brain tissue. And some of it is going to get diffracted and go back out, and some of it is going to get diffracted and go through the detector. And so this light that goes through the particular region here is going to get detected. It's going to measure the changes in oxygenated hemoglobin, and which will absorb the wavelengths of light differentially. So this is the shape we call a banana shape of light, and it's going to depend on the light path, and it's going to depend on distance between the emitters and detectors. So as we move the emitters and detectors further apart, we can get different regions and different depths of measurement. All right, so this was a quick summary. This is an overview of the absorption curves of hemoglobin, deoxygenated hemoglobin in blue and oxygenated hemoglobin in red. And you can see that they have different absorption curves for light at different frequencies. So that by having a measurement point below the isospecific point and one above, we can then do some fancy math. This is the modified Beer-Lambert law, that when we solve that and a few other equations, we can get measurements of changes in blood oxygenation and oxygenated hemoglobin and deoxygenated hemoglobin. We can also measure the total blood volume. So I won't go through the math today, but it is well published and documented. This is not things that we invented. Here's an example of an fNIR measurement. Here in the gray bar, the person is asked to do a motor task, typically a finger tapping or a hand clenching, and you can see the average oxygenated hemoglobin goes up after average over several trials. Here's a single trial and the dotted, and conversely, you can see the deoxygenated hemoglobin concentration go down. So what is the innovation that we brought to fNIR? Ourselves, we didn't. What we did is we partnered with a company called Arcunetics that had developed these really amazing LEDs that are very, very bright and stable. So here's an illustration of the transmission of their LEDs. And you can see typically LED. So when you're doing fNIR, you're flashing the LED at the 3 different frequencies. So one below the isospecific point, one at the isospecific point, and one above. And you flash them pretty quickly so that you want to get several measurement points pretty close to each other so you can solve these Beer-Lambert equations we talked about. And you can see that this typical LED would be in the one illustrated in black. Because it flashes, it's going to, by the time it heats up and lower and cools off, you get these sort of spikes, and they don't reach very high. Whereas their LEDs were very bright, very fast, and stay stable, and then go back down. So you get what you're going to be, the detector is going to have a lot more time, really highly activated to see some more activity. So we're going to be integrating the area over time, and that's going to give us a lot more signal than these sharp spiking. And we also had some custom silicon photodiodes developed with the gain built into them that provides really low noise amplification to the signal because it's being amplified right at the source. So that was a quick introduction to fNIR. Just trying to keep us on track over here. Sorry, you'll get to ask questions in the end, so please start writing your questions in the chat if you have them. Now let's talk about the integration. So what did we do here? So I described to you our EEG sensor. I described how we put a Faraday cage around the active electrode. What we did for integrating fNIR is we expanded that Faraday cage. So this Faraday cage here now just got made wider, and we put the emitters and the detectors inside it. So that protected them and their circuitry from the EEG. there's this spiking of the fNIR is going to produce artifacts, which I'll show you in a second, and we needed to protect them. So we put them inside the Faraday cage, and this allows us to isolate the electrical components. We also arranged them around the EEG. So EEG is in the center, and the emitters and the detectors are arranged around it in this arrangement. And this allows us to measure the path of light at the same location where the EEG is being measured. Of course, EEG is measuring from a broader area that's sort of summing into here, but at least you're co-localized measurement at the same time. We put these sensors through what we call these big pods, and those pods are spring-loaded. That allows the pods to adapt and adjust to the head. This is what these pods look like, and when they go on the head, they can adjust and comply to the via this spring that's inside them. So this is what they actually look like. Now, the other advantage of having this array of electrodes is we're able to do Mayer wave artifact reduction. So what is the Mayer wave? The Mayer wave is a systemic oscillation in the blood that's happening throughout our body, and it's going to affect, it was discovered to affect the fNIR measurements recently, and so that become a big interest in how can we remove it. Well, it's a systemic measurement. So if you remember, if we measure the short distance between the emitters and detectors, we're going to get a low depth path. So this measures just the skin, and we can measure the Mayer wave artifacts in the skin. And the long distance path is going to give us a measurement that goes through the brain tissue, as well as the measurement of that Mayer wave. But by having both the short distance and the long distance, we can do a linear regression and remove the artifacts from the long distance path, so we can regress it out. So this was some work that was here's some references for some of this early work that was done to develop these methodologies, and here's an example extracted from those papers where you can see a recording from the short distance path and the long distance path, and when you apply the adaptive filtering, you can get a nice recording here. So this recording here in the black traces is where you have an activation task, so maybe a finger clenching, and you can see the activated oxygenated hemoglobin, and then they stop the task, and then it goes back up and down as you activate the tasks. So having an array of electrodes allows us to do this at each site immediately. So it's a built-in feature. We have an emitter and a detector near each other, so we can get this short distance path, and then we can also get lots of long distance path. That also allows us to select the optimal path. If the longest distance path isn't bright enough, we can choose a second one. If neither of them are fine, we can choose a slightly shorter path. So there's several paths in each electrode that we'll be able to select. Oops, I'm not sure what happened in this figure here. It looks like it blew up on us. So this is the raw sorry, forgive me about that. So this is the raw tracing on the original one, and you can see that the lines are going quite a bit up and down. And here, and once we do the adaptive filter, the scale, sorry, I'm just going to adjust that so that we can see what we're talking about. Okay, so I apologize about that. But this allows us to, if we have a threshold, now you can see the artifact is on the more stable, and we can, by applying a threshold, we can more reliably determine whether we are above a threshold or below a threshold, as opposed to before the Mayer wave removal, we might be below or above, and we might be misled. So this was the illustration of another advantage of having this nice array. Now we need to talk about making sure that there's no interference between the two technologies. So here, what I'm going to show you is a recording of EEG, and the blue line is where the LEDs are off. So the fNIR LEDs are turned off, and then when we turn them on, you see the spiking of the LEDs getting turned on and flashing quite quickly. And that will introduce a big artifact if we disconnect the shielding. So when we disconnected the shielding, you can see these big artifacts coming through. And when we use the shielding connected properly, we see that there's no artifact even when we turn on the LEDs. So that is really critical, otherwise you're going to have to filter out all these artifacts from your EEG signal somehow. In this case, we don't have to filter them out because we shielded them, we protected ourselves from having them coming into the signal. Of course, conversely, one could ask, well, does the EEG have an effect on the fNIR? One wouldn't expect it. Still, this little experiment here demonstrates that. Here we had the EEG signal removed, the EEG sensor disconnected in the middle of the experiment, and then you can see that the fNIR signals, in the various frequencies, were not affected. They still have the same distribution even when we took off the EEG sensor. So the EEG doesn't affect the fNIR doesn't affect the EEG. That's critical to demonstrate and to have in an integrated system. So this was describing the pod, how the pod works. Each pod has 4 emitters, 4 detectors, with the EEG in the center. Each headset has 8 pods. We set up these pods, they're quite large, so we set them up, 2 on the front, 2 to get the prefrontal cortex, 2 on the temporal areas to get the auditory cortex, 2 on the motor cortices, and 2 in the back to get the occipital and visual cortex. So this headset was designed to, again, be easy to put on. All the electronics are embedded, so just like our EEG device systems, you can get up and walk with it. It has the built-in Mayer wave removal in the software. The data of the EEG and fNIR are being digitized by the same processor, so they are synchronized by default. There's no need to time align and pulse talk. And the fNIR will measure the oxygen hemoglobin, while the EEG measures the postsynaptic potentials of neuronal firing. So we're measuring 2 different physiological brain activities in the same device. This is what the software looks like. I'll show you that in a second, so I'm going to skip over the slide, and let's show you what the signal looks like. All right, so here we'll show a dawning. And you can see that there are some adjustment points. We will help the subject adjust them. So we're tightening, and there's another adjustment point that allows us to tighten the headset down onto his head. And then we can position the front hub, the top hub, so it's centered. There are here the ear clip sensors. And then we can attach the power cable, and then we work the sensors through the hair. I'm going to speed through that. So we adjust each of the sensors through the hair. And when we're done, we can check the impedances just as we did earlier, and then we cover the headset. So this is this little cover, and we're developing a prettier version of it. Protects us from the ambient light in the room. And then we have an embedded signal checker, so we will run that test. And that will give us the measurement of if there's a light leakage from the external and whether the light intensity is sufficient for the measurements we're trying to do. In this case, we got that the sensor T4 had some light leakage, so the subject is adjusting that sensor using the tool to make sure that it's sitting more stably on his head. And then we can rerun the sensor checking, and then we can do a tuning. So we have an automated tuning function that allows us to tune the system and get the optimal light intensity to get measurement that's sufficiently bright and not saturating the amplifier, the detectors. Then once we have that, the auto tuning is done. That takes about 2 to 3 minutes. Then you can look and individually check each of the signals. So here's the FP2, and we checked C3, and they both have really good pulsatile signals showing us that we're getting good measurement. You can, of course, go in and manually change all the settings you want if you don't trust the auto-tuning or if you want to optimize it for a different application. When you're done, you can go into the viewer where you can see the EEG data simultaneously with the fNIR data. So here was a blink in the EEG, and here's the fNIR data. You can view the oxygen hemoglobin, deoxygenated hemoglobin. You can activate the Mayer wave removal or not activate it. All the data can be saved and output raw. You have also the ability to reset the baseline, which is an important aspect for fNIR since we're looking at changes in the oxygenated hemoglobin. All right. So once we take this data, we can plot it in whatever our favorite signal and processing software is. So here where they had asked the subject to do some motor tasks, finger clenching, and you can see the activated area was getting these stereotypical responses activated when we're measuring, looking at the pod that's at C3. Of course, when we gave him a visual task, a checkerboard task, we could see that there was no such responses happening in relation to the activation area. So this is just a way to verify that the system has the selectivity that we expect, that the pods are responding to the tasks that are activating the specific brain areas above which they're sitting. This does have the Meyer wave artifact applied to it. Okay, one quick introduction to cognitive gauges, since we mentioned that in this device we extracted we placed the sensor pods on different cortical areas. The purpose of that was to measure mental workload in different areas of the brain. So we have an algorithm that we talked about last time. We'll have a separate webinar on that, but it's basically a machine learning algorithm that can take in all these different physiological sensors, EEG, GSR, fNIR. It applies a partial least square. It learns a model, creates an individual normative model quite quickly, and then provides an output. So in this case here in this video, we trained the model for mental workload and one for engagement. So this subject is engaged in a task, and you can see that his engagement is high. The task is pretty easy, so he gets it done pretty quickly. The mental workload is low. You can watch this video in a previous webinar, and there will be another webinar about it, so I'm going to skip over that for purposes of time. But just to say that we have this gauge that can take these physiological measurements and produce mental workload or other state assessments from these physiological measures. So in the case of the fNIR device, we did some visual, auditory, cognitive, and psychomotor tasks. It's very important, those tasks to be very well controlled if you want to show the selectivity of each of those. So for example, the visual task is purely visual task, so it's a visual discrimination task. The auditory task, the person is staring at a blank screen. During the psychomotor task, they cannot see their hands are behind a sheet, so they are not seeing their hands, they're staring at a blank screen. They're doing some different manipulations with their hands. In the cognitive task, the screen again is blank, and they're doing mental arithmetics inside their heads, counting up or down. Doing that, you're able to show that we can indeed have a visual response that's selectively showing a high activation of the brain when you're doing a visual task or auditory task, respectively cognitive and psychomotor tasks. So again, it's important. This is an example of what Q-states can do from this data, but you really have to be careful about how you control your task. Here are the specifications of the device. You're welcome to email Cameron or myself for getting a spec sheet of the device. Lastly, we'll talk about the applications. There's a very nice review paper published in 2017 that goes over a number of applications of combined EEG and fNIR, some of those covering devices, third-party devices that are built by other groups, some themselves, some commercially purchasing different EEG and fNIR devices and trying to integrate them together. The most exciting part that you cannot do without the combination of those is study neurovascular coupling. So that's looking at the relationship between the EEG and the oxygenation, whether that be for stroke, stroke rehabilitation, hypoxia, studying sleep and oxygenation during sleep, or epilepsy, surgery, anesthesia. So there's a number of applications listed here, as well as looking at the effects of stimulation, TDCS, DMS, and looking at the relations between the oxygenated hemoglobin and the EEG. Some people are also using those for enhancing the brain-computer interfaces by having redundant or complementary signals, provide error measurements or different state measurements. And of course, the question is, what can you do with it? And so we'll hope that you'll reach back out to us and discuss that. So the take-home message for you guys, I hope I showed you guys some high signal quality for both EEG and the fNIR, some devices that are motion electrical artifact resistant. They're really easy to set up. You'll have to trust me for now on about comfort, but please try us out. Come and have a sample or arrange a trial for you. We talked briefly about cognitive states, but all of this can be done for both EEG and fNIR in a real world or virtual world. So before I open up the floor for your questions, I want to thank you very much for taking the time to join us today. We've had a number of webinars so far, really well attended, and we'll have a few more coming up in the upcoming months. We'll have one about biomarkers and one about BCI, and we'll try to keep them going. So please send us your suggestions. All right, questions?
All right, thank you all. As Wally was mentioning, feel free to send your questions in the chat or in the Q&A box. We do have a couple that have come in. The first is, do the fNIR's diodes come in contact with the scalp? If not, how far are they from the surface?
Absolutely, they do come in contact with the scalp. So there is a conduit between the emitters are inside, but there's a conduit that brings the light to the surface, and that is making contact with the scalp and through this spring loading mechanism. So the redundancy helps. If one of the emitters isn't making good contact, hopefully enough of the other ones are, so that you can get a good reliable contact signal through. If it's lifted up, either the emitter or the detector, you get a lot of leakage of light, and that can cause saturation on the amplifiers, which is why the tuning can then come in and shut that one down, as long as you have another one that's working well in that pod.
Awesome. The next question is, what is the diameter of the pod?
The diameter of this pod, so I would say this is about 5 centimeters in diameter. The distance between the emitters and the detectors are just above 3 centimeters. I think it was 3.5. So the longest one is 33 millimeters between the longest path, and then we have some paths that are 23, we have some that are 29, 0.6, and we have the short one is 10.
Next question is it possible to do EEG and fNIRs when using TMS?
Yes, it is possible. It depends on which TMS device you're talking about. So the small TMS coils, and forgive me for not knowing their exact name, that can be handheld or sort of small cubes, those are fine. Those do not produce any risks. The large TMS coils, those can produce very large magnetic fields, and we have the Faraday cage that could have some significant heating, so we don't recommend using the large TMS coils with a device. You can do TMS before or after, since the device is easily removed and easily put on, so you can quickly get measurements before or after the TMS, but not with the large coil TMS. We can, of course, do TDCS, DECS, so we have integrated with the products from Soterex Medical, and you can do simultaneous TDCS or TACS simultaneously with those measurements.
Awesome. Next question, is it possible to compare dry EEG and fNIR on any kind of source reconstruction approach?
Oh, great question. Thank you for that. So typically to do source reconstruction, you need at least, most people agree that at least 19 electrodes, preferably more. But with 19 electrodes, you can do S-Loretta, or you can do BESA, source localization, and a few other methods. But however, with this headset that we only have 8 channels, 8 sensors due to the size of it on the head. So we don't have enough channels to do source localization with this device. We are reaching out to some groups that are developing some interesting software. For example, the group at USC is developing Brainstorm, and we're hoping to work with them to see how we can use the their analysis software, which is a very nice software platform that brings in multimodal data from MRI, fMRI, fNIR, MEG, PET, all sorts of data. They bring it in and they do sort of this visualizations and 3D reconstruction. So we're hoping to see what can be done with the 8 channels that we have. I don't want to promise you that you can do source localization with that few electrodes. It will be very, very coarse, but you can certainly get some measurements that are localized and try to do some interpolations and see what you can get.
Next question, have you observed any systematic differences between dry and wet electrode measurements?
Okay, yeah, great question. So we showed the figures that I showed earlier. We're showing you how well we're getting. We're getting 90% plus correlations between simultaneously recorded wet and dry electrodes. Of course, those are juxtaposed, they're side by side as opposed to co-localized like the fNIR and EEG. So that probably hopefully explains the differences. We do see at the very low frequencies, you start to see an increase in noise in the dry electrodes. We tend to see that that's within the variance. So sometimes you'll have the dry electrodes be slightly noisier. Sometimes you'll see the wet electrodes be slightly noisier. On average, the dry electrodes are slightly noisier, but we don't see a significant, it's not statistically significant, so it's well within the variance. Certainly between 0.1 and above, we're quite comparable to what the wet signals are getting.
So the last question that we have right now is, can we incorporate ERPs such as E' into the measurement system?
ERP, great question. So evoked response potentials, I showed some traces in EEG, and there's some people interested in doing that. For fNIR, of course, the time scales will be very different. Yes, the system does allow you to do evoked response potentials. We have a wireless trigger hub that can receive inputs from photodetectors if you put them on the screen for triggering, audio inputs for triggering, or push buttons. You can also send the signals using E' through the parallel port or USB. There's a nice adapter developed by Neurospec that allows you to get the USB into the parallel port. All of those go to the trigger hub, which gets wirelessly sent to the headset and provides triggers for synchronization with either E' or other stimulus presentation software like SuperLab or Presentation. All of those will work nicely and produce synchronized triggers. You can also use that trigger hub to synchronize with other devices such as the eye trackers or motion trackers, etc.
Let's get another question come in. What impedances do you get with your dry electrodes and what do you aim for?
Great question. So we typically get somewhere between 500 kilo ohms to a mega ohm, and that's what we aim for. We're fine up to 2 mega ohms. We prefer not to go above that. Really, you have to be, when people usually are doing wet electrode, they're used to talking about 5 kilo ohms and 10 kilo ohms. And we're at a much higher range. And so again, this is only possible because we have the ultra-high impedance amplifiers right at the source, and we have the Faraday cage shielding the sensors from the pickup of environmental artifacts. So we can do it because of a lot of mitigations that were put in place and a lot of custom circuitry that allows us to do this.
Awesome. So it looks like those are all of the questions that have come in so far. If anyone has any other last-minute questions, feel free to send them in right now. Otherwise, you're always welcome to send in your questions to either info or sales@wearablesensing.com. We're always happy to schedule a follow-up call to discuss any of these products in more depth, or provide a live demo and set up a trial, anything like that. So, yep, if anyone has any questions, feel free to send them in. Otherwise, I'll hand it off to you, Walid, to end us off.
So I just want to thank you all. It's hard not to see everybody in front when giving the webinars and interact with you that way, but thank you very much for your attention, and we look forward to seeing you at the next webinars.
All right, thank you, everyone.
Bye.